Great Salt Lake and its internationally important wetlands are but a small remnant of Lake Bonneville, shown here in light blue in the map on the left. Up until recently, in geological time, Lake Bonneville covered much of what is now the state of Utah. This pluvial lake was about the same size as Lake Michigan is today. Great Salt Lake is but a small remnant of Lake Bonneville, as shown here as a dark blue area in the map at left and in the satellite image on the right. Great Salt Lake is the fourth largest terminal lake in the world. Great Salt Lake's wetlands, along with their freshwater tributaries, are designated as a hemispheric site within the Western Hemisphere Shorebird Reserve Network. This designation signifies the vital importance of their habitat complexity and function, necessary to support millions of migratory and resident waterfowl and shorebirds annually. Here is a Google Earth satellite image showing the diverse habitat that occurs within these wetlands. Many of these birds migrate from breeding grounds as far north as the Arctic Circle to wintering areas as far south as Argentina, and then back again. Great Salt Lake wetlands are a life-saving pit stop along their journey. Birds depend on Great Salt Lake wetlands and open water for foraging, staging, breeding, brood rearing, and molting. North Davis Sewer District's wastewater effluent provides 20 to 22 million gallons of nutrient-rich water per day into the northeastern portion of Farmington Bay, Great Salt Lake. This creates about 1,200 hectares of sheetflow wetlands. These wetlands provide habitat for thousands of migratory and resident bird species. North Davis Sewer District and Jacobs Engineering have initiated plans to divert these waters away from the wetlands and into highly saline sections of Great Salt Lake due to concerns of Utah Division of Water Quality that nutrient loads from the discharge could cause harmful algal blooms in the wetlands or in Farmington Bay once these waters reach the bay, and could negatively impact recreational use. However, these concerns have not been substantiated by scientific data collection and analyses. Because of its uniqueness and wide diversity of habitats, Utah Division of Water Quality has designated Great Salt Lake its own beneficial use protection class, divided into 5 subclasses that include wildlife protection of a quality sufficient for waterfowl, shorebirds, and other water-oriented wildlife, including their necessary food chain. Consequently, North Davis Sewer District and Wasatch Front Water Quality Council have tasked Helix Ecological and researchers at the Council with collecting and analyzing scientific data to evaluate ecological interactions and nutrient dynamics within the food web in these wetlands to help determine the most appropriate management strategy. We delineated the wetlands into 6 units shown in yellow in this satellite image and installed 7 bird exclosure cages labeled as orange pins in February-March 2020. The numbering of the units followed the flow of the water from the treatment outfall to Farmington Bay, east to west. The amount of wetlands covered in water varied throughout the year, with most of the study area outlined in red having water in late winter, early spring, but much less in late summer, early autumn. Wetlands are approximately 3,000 acres or 1,200 hectares and almost completely dependent on treatment facility water, which enters the wetlands at a rate of about 20 million gallons per day. Here are the main research topics we are conducting in the wetlands in 2020. Chemistry and nutrient dynamics, water quality, bird counts and dynamics, bird macroinvertebrate, food web dynamics, invertebrate diversity, benthic algae and phytoplankton sampling on a regular basis, zooplankton assemblage, aquatic vegetation percent cover and biomass, and plant diversity and inventory. For a detailed description of field and laboratory methods used in our study, see the following link to our YouTube video. To our surprise, we found that these wetlands were much more seasonally and spatially dynamic than we anticipated. Here are graphs of predicted concentrations of 4 chemistry variables derived from linear and negative binomial regressions. pH increased significantly downstream from the treatment facility shown in Figure A. Ammonia was significantly higher in the central units, typically where there were more bird activity as shown in Figure B. Volatile, suspended solids and total dissolved solids were significantly higher at the junction of wetland water with Farmington Bay water and in Unit 4A shown in Figures C and D. We need to determine why 4A had higher concentrations of these two variables. Nitrate concentrations were highest downstream. TKN was highest in mid-reaches of the wetlands, and TKN-TN was mostly similar throughout. Most notably, orthophosphate concentrations significantly decreased as treatment facility waters moved through the wetlands towards Farmington Bay. Mean ortho concentration was about 5 milligrams per liter near the outfall. By the time water reached Farmington Bay, labeled here as Site 7, mean concentrations were at 0.04 milligrams per liter. This is over 2 orders of magnitude. Mean orthophosphate concentrations were below Utah Division of Water Quality's goal of 1 milligram per liter total phosphorus by the time the water reached Sites 5 and 6. Even in late February and early March, vegetation was growing in the slower water. Much of this was green algae that attached to the previous year's senescent vegetation. From late summer through autumn, aquatic vegetation prospered. The short emergent vegetation is a bulrush species. Duckweed and Stauchinia were also abundant, as was some green algae. Bulrush seeds, Stauchinia drupelets, and duckweed are important food resources for waterfowl, especially when invertebrate abundances are low due to constant consumption by birds throughout the summer. Patches of aquatic vegetation provide shelter for macroinvertebrates from bird predation and allows them to reestablish their populations. As water flows from the wetlands and enters Farmington Bay, it forms a multitude of channels and has lost much of its nutrient load. Slower water allows benthic algae to accumulate, which provides habitat and food resources for midge larvae and other invertebrates. Waterfowl tracks in the shifting sand reduce flow and provide microhabitat for benthic algae to grow. Canada goose feces in the upper center photo also reduce flow and provide both microhabitat and nutrients for benthic algae. Patches of benthic algae created by wading birds also provides superior habitat and food resources for midge larvae to grow. Water depth in this photo is about 1 inch. By late August, early September, bare, mostly sandy substrate gave way to thick patches of aquatic vegetation. Here is a photo of our bird exclosure cage number 6 amidst the growing aquatic vegetation. Much of the area surrounding the wetlands was dried mudflats or playas. Mudflat areas varied throughout 2020 depending on water level. Mean standing crop dry weight biomass of submerged aquatic vegetation, SAV, throughout the units was 195 grams per square meter towards the end of the growing season on October 6th. Standing crop biomass of benthic algae was 21 grams per square meter. However, benthic algae has an extremely high turnover or primary production rate, which can likely be measured in hours, particularly given the high predation of the algae by midge larvae, which we will discuss in a little while. We did not measure or witness any cyanobacteria blooms in these wetlands, although a nodularia bloom occurred in nearby sections of Farmington Bay in August. To paraphrase fish surveyors, counting birds is just like counting trees, except birds can hide and can fly away very, very fast. Check out our YouTube video link to test your skills at bird counting. Often, over 10,000 waterfowl and shorebirds utilize these wetlands daily. We don't know how long each individual bird remained in the wetlands as they are migratory and birds can fly. If each bird only remained for a week, then we conservatively estimated that more than 300,000 individual birds likely rely on these wetlands annually. This image is about 1/10 the area of open water in the study area wetlands. Given the difficulties of surveying birds, we estimated that there were roughly between 4,000 to 14,000 waterfowl and shorebirds using these wetlands, with as many as more than 16,000. Relative abundances of waterfowl and shorebirds varied seasonally, with percent shorebirds higher in spring through midsummer, and a higher percentage of migratory waterfowl occurring in late summer and autumn. Our findings also show that many shorebirds remain summer residents. Avocets comprised about 60% of the shorebird species that we observed throughout the year. Phalaropes, white-faced ibis, black-necked stilts, and other unidentified peeps made up the remainder. However, as with most of our bird count observations and analyses, observability was highly variable, and values that we present, although likely accurate, are likely not very precise. The basic food web of the wetlands is illustrated here, starting with producers, the plants. Consumers 1 are invertebrate grazers, filterers, and collectors that feed on producers. Consumers 2 are invertebrate predators such as dragonflies and damselflies that feed on consumers 1. Consumers 3 are waterfowl and shorebirds that feed on consumers 1 and 2 as well as the producers. Producers are dependent on the chemistry of the wetlands, particularly nitrogen, phosphorus, temperature, and salinity. The decomposers are mostly bacteria, and all the groups interact and contribute to detritus. Of course, the food web is much more complicated than that illustrated. Here are the major players in the food web of the wetlands illustrated in the previous slide. Go ahead and pause this video to examine the many major players. Our most important finding was that the ecology of these wetlands consists of mutualistic positive feedback loops between the consumers and the producers. These insights were edified by Heron et al. 2017 in the journal Ecology. The fuel, or energy, needed to initiate and propel this dynamic system are the nutrients from the outfall of the treatment facility, shown in the upper portions of the photo. Once the feedback loop is initiated, food web energy is typically measured as carbon transfer or biomass transfer. Midges are in the family Chironomidae and are often called chironomids. Larval taxonomy below family level is typically only taken down to subfamily or tribe. However, experts can identify midge larvae to genus and on occasion species. Larval mouthparts are required for taxonomic ID. In Figure 1, the striated ventrometral plate distinguishes the tribe Chironomini, of which the genus Chironomus are a member. The mouthpart, known as the ligula, shown in Figure 2, distinguishes the tribe Tanitarsidae, of which Tanipus neopunctipedis is a member. The red midge larvae, shown in Figure 3, are Chironomus, collected from a 0.1 square meter sample from the wetlands in May 2020. The first positive feedback loop in the food web, and perhaps the most important, is the mutualism between consumers 1, benthic invertebrates, particularly midge larvae, and the producers, benthic algae. Midge larvae were by far the most dominant benthic invertebrates in the sheetflow wetlands as measured by density and biomass. Two species of midge dominated, Chironomus species and Tanipus neopunctipedis. Densities of these midge larvae typically approach 20,000 per square meter and greater than 6 milligrams per meter squared dry biomass in the early part of the year. Vast amounts of midge biomass is needed to support the thousands of shorebirds that rely on this energetic food resource. The positive mutualistic feedback loop is essential. Heron et al. 2017 showed that as midge larvae densities increase, algal production increases, and consequently larval growth rates increase as their densities increase, likely because of the increase in algae. Without this positive feedback in the first loop, far fewer birds would be supported by these wetlands. Midge larval tubes were easily observed in the wetland soft substrate. Holes in sediment are midge larval tubes shown in the three photos. Inside the bird exclosure cages, larval tubes were uniformly distributed, suggesting intraspecific competition for space. Midge larvae tubes were less dense and less uniformly distributed outside of the exclosure cages, where shorebirds ravenously fed on larvae. The bottom image shows the three-dimensional structure of the tubes, which alter sediment chemistry, structure, and function. The second part of the mutualistic positive feedback loop is comprised of birds, their feces, and nutrients. Bird feces provide several tons of nutrients annually that are directly usable by benthic algae and aquatic vegetation, including soluble reactive phosphorus, SRP, and ammonia, NH3. Benthic algae are in turn available as food resources to the remaining midge larvae that haven't been consumed by the birds, which are then available as a food resource for birds. Of course, this is just one part of the complex food web of these wetlands. Early seasonal results from our bird exclosure cage study showed that bird predation significantly decreased benthic invertebrate density shown in Figure A and biomass shown in Figure B. Benthic invertebrate biomass was 2.5 to almost 6 times greater inside the cages than outside in spring, as showed in Table 1. Preliminary results for summer and early autumn suggest that biomass inside and outside the cages became more even, which we initially are attributing to the effects of increases in aquatic vegetation and subsequent changes in habitat and the invertebrate assemblage. Of course, midges don't only exist as larvae in the wetland sediments. After growing through 4 larval instars, midges undergo energy-intensive metamorphosis as they transform into aerial adults. Midges then emerge synchronously en masse after a very brief pupation period. Adult males, shown in this photo, typically emerge first and use their large fuzzy antennae to detect females. They then mate in large aerial swarms. Adults only live to reproduce and typically survive for less than a week. During this time, they are a major food item for predatory birds and superabundant spiders that inhabit the shores of the wetlands. Here is a photo of adult midges swarming along Antelope Island Road in October 2020. The road is popular for bikers who often wear face masks to keep from inhaling a few too many midges. After mating, female midges fly back to the wetlands to deposit their eggs and renew their all-important cycle in the wetlands food web. We also conducted an aquatic invertebrate inventory throughout the season. We used custom-made bug traps deployed in our Unit 3. Results are pending. We enlisted Blake Wellard, a Utah wetlands native plant expert, to spend one day in September with us inventorying plant diversity in and around Unit 3. Results are also pending. These wetlands are in nutrient and bioenergy hyperdrive, due in part to the mutualistic positive feedback loops fueled by nutrients from the North Davis Sewer District outflow. It is possible that these wetlands are seasonally nutrient limited in the farthest downstream units approaching Farmington Bay. We did not observe any cyanobacteria blooms in the wetlands in 2020, and we don't expect to see them occur in the future. Wetland waters could help dilute nutrients in Farmington Bay and potentially reduce cyanobacteria blooms in the bay. These wetlands are instrumental in meeting Utah Division of Water Quality beneficial use designations for the Great Salt Lake. They provide important habitat for thousands of waterfowl shorebirds and other wildlife dependent on this ecosystem. We have presented but just a few of our findings in this presentation, and we are just getting started on analyzing and understanding the ecology of this unique food web. We are developing complex quantitative food web nutrient dynamics models for these wetlands based on this past year's and next year's research. Several questions also need to be answered to better understand ecological effects of alterations to water flows by the treatment facility. The goal of our research is to provide scientific guidance on future water management options in the spirit of the Clean Water Act and to support water quality designated beneficial uses. Here are links to a few videos we made during our research. We gratefully thank the following, and we thank you for watching our video.